Hello, this is Professor Matt Katroulis of Rio Hondo College. This is my video lecture for Chapter 16 on amino acids, proteins, and enzymes for Chemistry 110. When we talk about proteins, which are really the heart of this chapter, the way we can best describe them is as biological polymers, and they have a number of roles in our bodies and throughout organisms. But perhaps one of the most important is their ability to facilitate chemical reactions within an organism. Being polymers, that means that they are made up of smaller units, just like, for example, our polysaccharides were made up of monosaccharides. But the units that make up the protein are called amino acids, and they're going to be the focus of the first part of this video lecture. So within a protein, we see that many amino acids are attached in a long chain, and the specific sequence of these amino acids and the way that those amino acids then fold will ultimately determine how that protein will behave in the cell and what function it has. So the individual units, being essentially composed entirely of non-metal atoms, will be linked together by covalent bonds. And the name of these bonds that occurs between the individual amino acids unit is a peptide bond. And we will take a closer look at the peptide bond in just a moment. For now, though, let's focus on the amino acids. There are 20 amino acids which ultimately end up in the proteins. And of these 20, we are able to synthesize exactly half of them within our cells on our own. But the remaining 10 are going to be called the essential amino acids. These are listed in the textbook, and it's not necessary that you memorize them. For at least 19 of the amino acids, their structure is going to be essentially the same. The 20th is a little bit different, although it's reasonably similar, as we might see, to the other 19. But they're going to have a carboxylic acid group bonded to a carbon atom, and we will call that carbon atom the alpha carbon. And then that alpha carbon will be bonded to an amine group, hence the name amino acid. We have a carboxylic acid and an amine. The alpha carbon, very importantly, is bonded to what we are going to call a side group. And it's the differences in the side groups that account for the diversity of different amino acids. Every one of those amino acids has its own unique name, and assigned to those is a 3-letter code, which is in many cases the first 3 letters of the name of the amino acid, although not all, and an even simpler code of 1 letter. Since the 1-letter codes don't often correspond in any way to the name, I don't like to use those. I will instead stick to using 3-letter codes when I do refer to them with amino acids. So here is our basic structure of what an amino acid might look like. So here we have a carboxylic acid group off to the right. This is our alpha carbon right here in the middle. It is bonded to a hydrogen and a side group. And then that is bonded to an amine group. So here is a set of some of the different amino acids. This first group you'll notice across the top, they're all identical except for the side group, which we have colored in here. So we've got glycine, alanine, valine, leucine, isoleucine. And you'll notice that all of these, importantly, have side chains that are made up of hydrocarbon groups, except for glycine, which simply has a hydrogen. And then we have several more down here, some of which contain sulfurs, others alcohols or carboxylic acids. And there are even more amino acids here. So you'll notice that some of the amino acids have acidic side chains, like we have right here. Some of them have amino side chains, like we see with lysine. Some of them are fairly complex, like we see with arginine and tryptophan down here, and histidine. One of these is fairly special, and I want to call it out, and that is proline. If you'll recall, I said that the structure was essentially the same for 19 out of these. Proline still follows the pattern of having a carboxylic acid bonded to an alpha carbon bonded to an amino group. But you will notice that with proline, there is a ring in addition to the bond between carbon and the nitrogen that connects these. And so it's a little bit different compared to the others. Now, with the exception of one of the amino acids, that being glycine, the alpha carbon is a chirality center in those amino acids. Why? Well, if we go ahead and take a look at, for example, alanine right here, you'll notice that the alpha carbon, which is shown here as part of a Fischer projection, is bonded to 4 different groups. So you'll notice that this is presented a bit differently than we saw a moment ago. And it's common to draw amino acids in different ways. But with this particular Fischer projection, we see that the carboxylic acid is drawn up and the amino group is drawn to the left. And you'll see that we have a hydrogen and we have our methyl group. That methyl group is the side chain for alanine. Why is glycine going to be special? Why is it not a chirality center? Well, if you go back and look up the amino acids, glycine would have a hydrogen as the side group right here. So if we have a hydrogen here and a hydrogen here, those would be 2 identical groups, and that would mean that this is not a chirality center. So with the exception then of glycine, which is not chiral, we have a possibility of 2 enantiomers for the different amino acids. And essentially all of the amino acids that we are interested in have the L configuration, as opposed to the carbohydrates, which were all D. We're not going to really get into the distinction between what makes this L and what makes this D in this class. That's kind of an advanced topic. But ultimately, if we draw them as a Fischer projection, this is how we would do that. We're not going to need to use Fischer projections nearly as often with amino acids, though, as we did with carbohydrates. There are occasionally found in nature some of the D amino acids, although that's not necessarily to say that they are encountered in proteins. Now, depending on whichever book you are reading about or the author's focus, you'll notice that they have many different ways of categorizing amino acids. You'll notice when I first talked about the amino acids, I was pointing out that some of the amino acids had hydrocarbon side chains. That's one way in which they can be categorized. But I'm just going to follow your textbook's convention of categorizing them into 3 categories. And the first category will be the acidic amino acids. I know that might sound obvious, amino acid being acidic, but what we're referring to here specifically is the side chain. So if the side chain has a carboxylic acid, this is in addition to the carboxylic acid that's already present in the amino acid, we will call that an acidic amino acid. There are 2 of these, aspartic acid and glutamic acid. That's another easy way to recognize them. They have the word acid in their names. We will say that the amino acids are in the basic category if they have a basic nitrogen atom in their structure. So this would include arginine, lysine, and although it's not going to be easy for you to see, histidine also has a basic nitrogen. And the rest of them, the other 15 amino acids, we are going to say are neutral. And as I had mentioned a moment ago, we have other ways that we can classify these. As I said, the ones that have nonpolar chains versus ones with very polar side chains, or those with aromatic side chains. So we can break them into many different groups if we chose to do so. At physiological pH, meaning essentially the pH that you encounter most proteins, which is fairly close to neutral, the amino group is a base, and it will always be found in a protonated form, meaning instead of as NH2, it will be found as NH3+, as I'm showing down here. And similarly, the carboxylic acid will always be deprotonated, meaning it's lost NH+. The reason for that is this molecule has both an acid and a base part, and so amino acids would ultimately react with one another in an acid-base reaction before they did anything else if they were combined. So this hydrogen would be ultimately transferred over to the much more basic nitrogen. And what we call these species is a Zwitter-ion, from the German zwei, which is two. So it is a structure here which has two charges in it. But overall, the molecule is neutral. And that will be the case for 15 of the amino acids, the neutral amino acids. The acidic and the basic amino acids will typically have an additional positive or negative charge depending on their side chain. So, for example, the acidic amino acids will generally have a negative charge down in their side chain, and the basic ones will have a positive charge there. And so this is just essentially showing this in greater detail. Aspartic acid, for example, you'll notice that we have the carboxylic acid deprotonated, we have the amine protonated, again meaning an extra hydrogen has been added along with a positive charge. And aspartic acid normally would have been CH2COOH. That hydrogen has been removed at physiological pH. Lysine, we have the opposite circumstance. If you notice here, we have a chain of 4 carbon atoms, and then another amino group, which would ultimately be protonated at physiological pH. Now let's talk about how these are combined. 2 amino acids are joined together in a very specific way. The amine group of one of them will join with the carboxylic acid group of another. And we eliminate a water molecule in this process. This is what we call a condensation reaction because it produces water. And so, as I said, we call these bonds peptide bonds. Ultimately, though, they are just going to be amides. If you'll recall from chapter 13, the combination of amines with carboxylic acids should produce amides under specific circumstances. Now, incidentally, if you just throw a bunch of amino acids together, they're not going to react because of the fact that the carboxylic acid is deprotonated. That makes it much less reactive. And the amino group is protonated, which makes it much less reactive. So this joining has to be assisted by various processes within the cell. Proteins, for example, are generally built on ribosomes, something you learn more about in biology courses, and ultimately the processes involved there help out in putting these amino acids together. So let's say here we had this set of 2 amino acids here. They may be the same, they may be different. We're just putting R here for their side groups. We combine these 2 together, and we kick out water, and we end up with a new peptide bond. And you will notice that this peptide bond is an amide. Since we have another carboxylic acid, this could continue with another amine, and this process could be repeated hundreds of times in constructing a protein. And so what we call this then is a polypeptide when we have them all combined together. You'll notice the pattern here of we're going to typically start with a nitrogen. That's going to be our convention, is to always start from the amine end, the nitrogen end, where it's possible to do that at the left. And then it will go nitrogen, alpha carbon, the carbonyl, you'll remember that used to be the carboxylic acid. Nitrogen, carbon, carbon with the double bond oxygen, nitrogen, carbon, carbon, double bond oxygen. And that pattern essentially just repeats over and over and over, with the R groups being the main things which differ as we go across the polypeptide. So as I mentioned a moment ago, we typically list polypeptides from left to right, with the amino side being on the left whenever we can do that. We call that left side the N-terminus because it begins with a nitrogen. We call the other side a C-terminus. It ends in carbon. Of course, people could draw it backwards. It's just that we are always going to interpret it by counting from the N-terminus here. And so we can essentially name these polypeptides or list them as three-letter codes, always starting from the N-terminus. And so let's say, for example, we had a polypeptide, and it had 4 amino acids in this order. So starting from the N-terminus, glycine, alanine, methionine, leucine. We can abbreviate that using the three-letter codes for each of these. So glycine, GLY. It's customary to use a hyphen here, although it's not always required. Alanine, methionine, leucine. And that's probably the most common way to list a polypeptide. It is also possible to name them, although this process ends up being rather messy. And the way we do that is we essentially just change the ending for all but the last amino acid in the polypeptide from its ending to il, YL. So with the exception, for example, of leucine, these would all be given il endings. So the name of this, and I've used color here to make it a little bit easier to see how these all break down, it would be glycyl, alanyl, methionine, leucine. Okay, so again, just chopping off the ends, glycine became glycyl, alanine, alanyl, methionine, methionine, and then leucine is left alone. Okay, what I would like to do here is look at this polypeptide, and first let's figure out how many amino acids are involved in it. Then let's give the sequence of three-letter codes that correspond to it, and then let's give it a name. So the first thing we want to do is figure out where the various amino acids start and end. So if we just start from the N-terminus, we have N, carbon, carbon with double bond oxygen. So that's our first amino acid. Let's separate it out there. And then the next one, N, carbon, carbon. So there's a second one there. And then this is the C-terminus. Technically, it does have oxygens at the end, but we still call it the C-terminus. And now what we need to simply do is look up each of these amino acids. So this amino acid here, with the phenol group on the side, is called tyrosine. This one is alanine. And then this one is cysteine. And so the three-letter code is TYR for tyrosine, alanine, cysteine. If we want to name it, tyrosine becomes tyrosyl, alanine, alanyl, and then we leave cysteine alone. So tyrosyl, alanyl, cysteine. I'm sure you can imagine that when these polypeptides have 100 amino acids, we're not going to bother naming them. Instead, you'll just simply refer to them by a name like subunit 1 or something like that. So now that we have our amino acids coming together, we want to go on to the next broadest level of looking at these concepts, and that is to look at protein structure. What happens when we get those amino acids together? So these molecules are incredibly complex in most cases. And so what we can do is we can look at a protein and 4 levels of it at a time. And so we're going to start off from the very most basic, most simple level to the most complex. So first, the first level is called the primary structure. We've already seen the primary structure of a polypeptide just a moment ago. All the primary structure is a list of the amino acids in order. And so it tells us ultimately nothing about what the protein is going to look like. But as I say here, it's essentially a recipe for building the protein if we wanted to do so. And it is possible to do this chemically. It's very, very tedious to do so, although there are special types of machinery that can automate this process for us. So this is a very pretty version of doing this. This would just be, leucine, methionine, etc. as we go through here listing these. Normally it would just be a 3-letter list like I just showed you a moment ago for a polypeptide. The secondary structure really focuses on 2 types of structural elements that we find within proteins. Most of the proteins that we deal with contain an alpha helix within them, and the term helix just describes a coiled region. We've already used the word helix before when we described, for example, starch molecules. And then we also find a very common set of structures called beta sheets, or sometimes called beta sheets or beta pleated sheets, which is where we see a peptide chain essentially folding across itself, basically like a sheet, in an almost flat plane, not quite flat, but reasonably so. And we find these certainly not in all proteins, but in many of them. Ultimately, what keeps these together is going to be hydrogen bonding from one subunit to another or from other elements within the structure of the amino acids. So here is an example of an alpha helix. You'll notice that these are many different amino acids, and we're not going to try to sit here and identify all of these, and we can't because you'll see that there are R groups for all of them. In all cases, the R groups are external to the alpha helix. They're pointing out. This ribbon right here is meant to really show you how this is a helix shape. If I took that ribbon away, it would not be at all obvious that this in fact is a spiraling kind of a shape right there. And ultimately there are hydrogen bonds, although it's not really easy to see them. But for example, here is a carbon double bond oxygen, and over here we have a nitrogen with a hydrogen. There could be a hydrogen bond right there to hold these together, and then another hydrogen bond from here, this oxygen, to here, this hydrogen. It's kind of hard to see in these 2-dimensional structures, but there's lots of hydrogen bonds between different parts of the helix. A beta sheet, on the other hand, is showing a region where we have multiple amino acids just essentially laying in a plane. In special types of drawings called ribbon diagrams, we usually represent these helixes as helixes. And then we will typically represent these sheets using large arrows that point in the direction of usually from the N terminus to the C terminus. The tertiary structure gets even more complex, and it's not really possible with the tertiary structure to give a name to the shapes in most cases because the shapes are so bizarre and amorphous. It's kind of like when you're trying to look up at the stars and name constellations, how you have to be so creative in coming up with shapes. It's essentially the same thing here. It's not usually easy to give a shape name. Sometimes people will call things fingers or barrels or something like that, though. So the tertiary structure is ultimately the three-dimensional shape of a polypeptide. Now, proteins can contain several polypeptides within them. So the quaternary structure would then be, how do those polypeptides then combine to form a whole protein? And sometimes there are also other molecules that participate in this process as well, or even metal ions, like for example zinc ions are common in some proteins. So the tertiary structure then is basically the total structure of an individual subunit, and then the subunits coming together gives you the quaternary structure. In just a moment, we're going to look at the types of bonding which impact the tertiary structure. And you'll notice that some proteins do contain only one subunit, in which case the quaternary structure would be the same as the tertiary structure. So this diagram is showing to us all four levels. So the primary structure, which again is just a list of the amino acids, we get no shape information about that. Then we have the secondary structure, which is the alpha helixes. You'll notice here that the hydrogen bonds are kind of nicely detailed in this particular picture. And the beta sheets. And then if we go ahead and we look at the entire list of amino acids here, and we consider them all and how they're going to fold and bend over into different shapes, that's going to give us the tertiary structure. So within the tertiary structure here, you can see it's kind of difficult to give an overall shape. I guess I could call this bean-shaped, but that, of course, is because this bean drawing has been drawn around it. Without that, it would be very hard to see. You'll notice that we have the alpha helix right here. You'll also notice that the beta sheets in this particular diagram are kind of represented by these little zigzags. As I mentioned a moment ago, it's more common to use arrows to indicate beta sheets. And then if this tertiary structure is not the end, in other words, if there are other polypeptides that comprise the protein, as in this case it looks like there are at least 4 structures that I can see, combining all of those structures together would give us the quaternary structure. Here is a vitally important protein called hemoglobin. Hemoglobin is responsible for transporting oxygen into the cells through the blood. And if we look at this, there are different colors to illustrate the different subunits. So if I was to look at the blue only by itself, that would be the tertiary structure of one subunit. And then if I was to look at the green by itself, that would again be another subunit here. You'll notice in this particular diagram that we have alpha helixes, so those are those twisting regions right there. If you'll also notice, they're not as easy to see, but there are arrows in places. So for example, we've got an arrow right here. That's another way of representing a beta sheet without showing all of the individual atoms. And then this connection right here is just essentially indicating that we have more peptide bonds throughout here. So this area I'm pointing here, this might be 5 or 6 or 7 different amino acids as we go from a beta sheet into the next region, which looks to be another beta sheet or possibly an alpha helix. It's really kind of hard to say with these diagrams the way they are. So you'll notice that there are 4 subunits here, so the 4 different colored regions. And in addition, there are 4 what I would call auxiliary molecules, so additional molecules that help out. And these molecules right here are called hemes. These are fairly complex organic structures which have an iron atom which is charged, embedded within the center. And that iron being charged can hold on to O2, oxygen, and help to transport it throughout the cells. This is a very interesting protein in that it changes shape depending on whether it's carrying oxygen or whether it's transporting away carbon dioxide. And it is the change in this shape, whether it is oxygen rich or oxygen poor, which ultimately is going to lead to the difference in the color of blood. So when it is transporting oxygen, it is a red color. Or when you bleed, regardless of whether the oxygen was red or blue before you begin to bleed, oxygen will immediately bind to those ions, and that would cause the blood to turn red the instant it's exposed to an oxygen environment. So let's see if we can look a little bit more in detail. And the focus here really is kind of on the tertiary structure of the proteins. Now, most of the proteins in the cell are surrounded by the cytosol, which is essentially the cell's solution, which is mostly made up of water. And so it would make sense that, given the choice, a lot of the amino acids which, like water, the ones that are able to hydrogen bond and have polar side chains, are going to tend to twist towards the outside of a protein. And we call those the hydrophilic regions, the water-loving regions. The nonpolar amino acids, the ones which have side chains that consist of things like methyl groups and isopropyl groups, things like valine and isoleucine, they tend to be encountered more often towards the inside of a protein, where they are attracted to other nonpolar amino acids and where they are essentially isolated away from the water solution outside. This is not absolute between these, it's just a tendency among these just different amino acids when they try to come together in the optimal shape. Cysteine, as you'll recall, has a sulfur atom. So does the amino acid methionine, but cysteine was special. It was a thiol. It had a sulfur with a hydrogen on it. And what we find happens is that the cysteines in different positions on a peptide chain or a polypeptide will tend to bond with one another to form what we're going to call a disulfide bond here. And so this is going to add a very strong interaction, an actual covalent bond right across the polypeptide, from one part of a polypeptide to another. So a cysteine bonding with another cysteine. And that is, as I said a second ago, called a disulfide bond. I'll show you a picture of that in just a moment. But if we think about this, because covalent bonds are much stronger than, say, hydrogen bonds, this is one of the strongest forces that can hold proteins together into a kind of a constant shape. However, they do tend to be broken rather easily if the protein is heated. Now, that's called denaturing of a protein. You can heat it, will destabilize the shape, or chemicals can destabilize its shape. But heating it to too high a temperature, like we do when we boil an egg, will cause those disulfide bonds to break. Okay, so for example, a very important structural protein is keratin. And if we look here, this is essentially an alpha helix forming one part of a polypeptide. Here's an alpha helix of a distinctly different polypeptide. And you'll notice that these polypeptides are being joined together by a disulfide bond. So this was originally CH2SH. It was cytosine. Same thing here. And so now, by forming this bond in between them, it is a covalent bond, which makes it stronger than most other attractions. But sulfur-sulfur is a fairly weak bond overall. So as I mentioned a moment ago, if it gets heated to fairly high temperatures, it will break. But we're not expecting to encounter temperatures of that magnitude within the organism. Let's take a more detailed look at all of the different important interactions which can occur within a protein. And although this diagram looks very complicated, in fact, all of these forces are forces that you should already be familiar with. So just a moment ago, I mentioned that the amino acids which are hydrophilic, the ones that can hydrogen bond, tend to project away towards the outside of a protein or a polypeptide. So if this is our polypeptide here and it has a very amorphous shape, you'll notice that we've tried to show that the hydrophilic side chains tend to be pointing out and away. Also, you will notice that there can be hydrogen bonds from one side chain to another, as we see here. So hydrogen bonds not only between the outside amino acid side chains and water, which would be surrounding this, but we can actually get other additional hydrogen bonds from one side chain to another, along the side of the protein, or actually even internally. On the inside of most proteins, we find again the less polar, the non-polar generally, or hydrophobic side chains. So for example, here I'm showing an amino acid. This was alanine, the one that has a methyl group here, wanting to be near another alanine. Here we have another one called phenylalanine, being near another phenylalanine. All of these are non-polar, and so they're likely going to want to be near another one. So when we look at some of the attractions within a protein, we have these kinds of attractions. What do we call those? Non-polar to non-polar? These are the London dispersion forces. So we have London forces here from non-polar to non-polar groups. Again, these are supposed to represent lots and lots of amino acids. And here, I must have a cytosine with another cysteine with another cysteine, and these are reaching across and bonding with a disulfide bond. The same thing here. That helps to maintain the shape here. And the other important attractive force that you'll want to be able to identify here are the attractions between ions. Remember a moment ago I said that the acidic side groups tend to be negatively charged in solution, and the basic ones tend to be positively charged. So it makes sense that there would be attractions from one side to another. These aren't true ionic bonds, but they are quite strong attractions. I believe your book calls these electrostatic interactions, which would be true. That's the attraction between opposite charges, although the term salt bridge is one that I actually prefer. I don't really like ionic bond because that implies the kinds of bonds that we find with metals and nonmetals. This is a little bit different there. But salt bridge is great because it's showing the attraction from a negative charge to a positive charge. So you'll want to be familiar with all of these types of attractions again. So the hydrophilic groups, which can form hydrogen bonds with the solution on the outside of the protein, hydrogen bonds which can form internally within the molecule, disulfide bonds, which are the only type of attraction which is not an intermolecular force. These are actual covalent bonds. And then our salt bridge here, which is the attraction between oppositely charged ions. Taking a more detailed look at sulfide bonds, we saw already disulfide bonds in the keratin proteins, which are in hair. And so ultimately, to hold these structural units together, we have lots and lots of disulfide bonds to keep them in their place. And so ultimately, it is the position of those bonds in relation to the keratins that determines the natural shape of an individual's hair. Now, as we all know, you can style your hair into different shapes. You can give it a perm by using various chemicals that can break the disulfide bonds. You probably never thought about it that way. You never went to a hairdresser and said, hey, I'd like you to break my disulfide bonds and reform them for me. But essentially that's what you're doing when you go to get a permanent. And so ultimately what we can do then is we reduce the disulfide bonds and then reoxidize them to form a new hairdo. And so this is the process that we have here. So I want you to notice that the way these are supposed to represent two different polypeptide strands between two different keratin molecules here. Excuse me, keratin. Not keratin, keratin. And so we have natural hair with its disulfide bonds. When you go to your stylist, they add a chemical which acts as a reducing agent. If you recall, we talked about a reducing agent. When something is being reduced, it is either losing bonds to oxygen or gaining bonds to hydrogen. And that's what we see here. We see that these hydrogens are being added across here. And now, ultimately, since these are broken, what can happen is you can reposition the molecules relative to each other. So when curlers are used and the hair is twisted, that repositions the disulfide bonds. And so you notice here that these pictures have been shifted to show that the positions have been changed somewhat. And then to get the disulfide bonds to reform so that the hair will take a new shape, we add an oxidizing agent. And now a permanent wave is there. Of course, that permanent wave only lasts for a time, because over time these bonds will break and the fresh hair that's coming out of the scalp will not have the memory of the curlers that the hair that was actually involved in the perm had. So this is, as we all know, not a permanent process. The final topic I would like to talk about, and in rather simple terms, is enzymes. So enzymes are proteins that act as catalysts within the cell. It is a fairly complex subject, but we just want to remind ourselves what catalysts are. We said that catalysts are chemicals which speed up a reaction, and very importantly, they are not used up in this process. And so what that means is an enzyme speeds up a reaction and can be used over and over and over to catalyze the same reaction multiple times. Ultimately, each enzyme will usually catalyze only one very specific reaction. And that's owing to the shape of the enzyme. So what it does is each enzyme will have a very specific shape, and the amino acids within that shape are positioned in a very special way that will help to hold different atoms together or pry them apart to facilitate a reaction. And so that will cause it to speed up that particular reaction. In all likelihood, though, the positions between those vary from protein to protein. So although there are similarities from enzyme to enzyme, they are often going to behave in very, very different ways. And so we're going to take a very simplified approach to this. And so usually what we see here is that chemicals that are going to be catalyzed become bound to the enzyme. So they are attracted to specific amino acids within the enzyme's protein, and they are held into place. The enzyme will then often twist into a shape which will put particular atoms either closer together or farther apart. And then with the atoms reoriented, that's going to make the reaction go much faster. Remember when we talked about chemical reactions, we said that for a reaction to occur, atoms have to be in a particular orientation. And so the enzyme is really facilitating that. It's helping to hold the atoms in the correct orientation. So, for example, here is one particular enzyme called sucrase. And what you'll notice here is that the purpose of sucrase is to take sucrose and break it down. So how does this work in a very simplified way? Well, the enzyme has a very specific shape, and there is what's called a receptor site. And the receptor site doesn't look nice and pretty like this with a hexagon and a pentagon, but we're going to keep it simple here. And so the site is designed so that any sucrose molecules which happen to bump into the enzyme will fall into that position, and then they become bound to the enzyme. So we now have sucrose, which is represented here. This is a glucose and this is a fructose, which are joined together in sucrose, and they are bound here. Then a water molecule in this particular case is added, and although you really can't see it in this drawing, the enzyme is slightly changing its shape, and that's to help facilitate the separation, the breaking of the bonds here between the glucose and the fructose. And then the product is released, giving us back our enzyme, and it still has this same cavity here, our receptor site, so that another sucrose can come along and fill it, and this will just go on and go on and go on. A lot of these enzymes also have other spots on them, which, owing to the presence of special molecules that they detect, can tell them to stop working or to start working again. So they literally have like their own little on and off switches. For example, like if fructose started to build up, there might be over here a switch that detects fructose, and if there's fructose held in it, it might say, like, hey, you got to turn this off. We don't need to break down anymore. That's a very general picture of how that works, and it's certainly not necessarily how sucrase works, but it's a very common type of mechanism for enzymes. So the most important thing to understand then again about the enzyme is that they tend to have receptor sites where molecules fit within them, and then the chemical reaction is carried out in the receptor site. We also often see enzymes that do the exact opposite of what we're seeing here. So this enzyme was designed to break two groups apart. It's also very common to have an enzyme that does the opposite, which is to say it takes in two groups and then joins them together and then spits them out. That is the end of our very broad look at amino acids and proteins. Thank you.